My name's Doug Bruce. I teach here at Laney College, and I teach in the biomanufacturing program. I wanted to tell you a little bit about the program. We train students to work in the local biotech industry. So if you know about the industry here, it's birthplace of biotech. We've had students placed at pretty much every company you could name: Genentech, Bayer, BioMarin, Novartis. And in the last 3 years, it's been all of these food tech and material tech companies that have risen out of basically thin air with IndieBio and accelerators in our area. So probably 70% of our placements in the last 3 years have been in food tech, so like Geltor, Perfect Day, Wild Earth, and the Celag type companies. So I just wanted to let you know what we're doing a lot of the work in CRISPR and synthetic bio now too that you'll hear about tonight. You're gonna hear about CRISPR. So if you want to find out more about our program, check us out. We're on the web, laney.edu/biomanufacturing, or just Google us. So we got some info back at the table there too. You can get on our mailing list and find out more about our program. So it's my pleasure tonight to introduce our speaker, Dr. Josiah Zayner. I pulled this from his company's website, the Odin, so easiest way to get that kind of info. Dr. Josiah Zayner is a global leader in the BioHacker movement, constantly pushing the boundaries of science outside traditional environments. He started biohacking during his PhD in molecular biophysics at the University of Chicago, creating the chromochord in his apartment, the world's first musical instrument that uses engineered protein nanotechnology. After his PhD, Josiah received a prestigious fellowship to work with NASA's synthetic biology program, engineering bacteria to help terraform Mars. Now he is the founder and CEO of the Odin. Josiah has a number of scientific publications and awards for his work in protein and genetic engineering, and is also recipient of art awards for creating speculative science works that have been featured in museums across the world. His work has been featured in Time, Scientific American, Popular Science, Business Week, The Verge, NPR, and among many others. He enjoys whiskey and Red Bull, sometimes together. And with that, let's all welcome Josiah Zayner to the front here.
So today's not probably going to be a traditional presentation. I mean, I've never been to one of the presentations y'all have here, but this is not going to be very traditional. I hope you all do learn something. We're going to talk about CRISPR a little bit, but in general, we're probably just going to talk about science, the way science is done, how I view science, and how I view the things I do. Because not to be negative or pessimistic, but I think that there's so much more science has to offer than the current way it's done. There's so much more that people can do. And I think it's just looking at it differently. It's thinking about science as something that you can express yourself with. Something that you can create something cool and beautiful with. To me, that's what the whole point of science has always been. And, that might be getting fruit flies high on cocaine, but it might also be just, making something that's completely insignificant to, medicine and technology, right? Our worlds don't have to revolve around blitz-scaling companies and, curing cancer. That's all right. I promise you. Anyway. So I changed the title of my talk a little bit. How you can cause a zombie apocalypse to how I cause a zombie apocalypse. Because, I get interviewed a lot by a lot of people. Even when, I used to take my company and apply to startup accelerators and things like that, and even when I interviewed at Y Combinator, the same question over and over again: is this going to cause an apocalypse? You know, is letting people do genetic engineering in their homes going to cause an apocalypse? And I'm just like, no, like, what? Stop asking that question. Like, come on. What is this hyperbole that you're throwing at me? You know, like, you're watching too many movies. So I thought maybe I could frame this talk in the way that my life has led me to create the zombie apocalypse. And through that, maybe I can teach a little bit about CRISPR. So I started out, I was terrible in high school. So I like almost failed out of high school. You know, for somebody who's standing in this position, that's, I think, pretty crazy, right? I got a PhD, I worked at NASA, I have my own company. If you look, my grades, what, biology, C, F, C, D, C, you know, I kind of work in biology now. It's pretty disappointing. I think my best class was, what, health education? Hey, you know, not too bad. World history I got a B in. And I think PE, how did I get a C in PE? I don't even, like, what do you have to do to get a C in PE? I don't even know. Like, what did I do wrong there? I don't even remember. It's terrible. I think I was ranked something like in the bottom quarter of my high school class. I was 600 something out of 800. Pretty terrible. I never, you know, I grew up in poverty. None of my parents had gone, neither of my parents went to college. Like, I had nobody around me who was telling me to go to college or who was in science or a scientist or anything like that. I didn't even think college was something that I wanted to do or that people should do. I didn't care, you know, I never imagined my life would be like it is now. So I just did what I wanted. A lot of that was just reading books and learning. I got into computers a lot, you know, taught myself computer programming and worked on that. But after high school, I was just like, I don't know. I didn't know what to do. That was complicated by the fact that I was arrested twice between the ages of 16 and 18. Once for assault. It was a fight in high school, don't worry, it wasn't that bad. And another for possession of marijuana, which was, you know, in the late 90s. It's crazy, you know, it's like legal everywhere now, including Illinois where I live. And yeah, I was busted for it. Less than a gram of marijuana I had on me. Gosh, that's terrible. Yeah, this picture is actually from Alcatraz. I went there once with my mom. It's not my mugshot. Sometimes I wonder if my mugshot's like online, you know, one of those websites where it's like, find out if he has a criminal history. But yeah, like, you know, my life was not set in motion for me to work in science at all or explore scientific things. It was set to go in a very negative, you know, direction. But I, you know, decided to go to community college, to Moraine Valley Community College near Chicago. And I actually did all right. And I got interested in biology and health stuff, and I transferred to a university and got a bachelor's degree in plant biology. I think plants are the coolest thing ever, but I can't take care of a plant at all. Like, I'm not even kidding. I have a degree in plant biology, and I kill all plants. I even tried to grow marijuana once, and I just destroyed it. I killed it, and then I put it outside, and I was just like, I'm going to let it die. And then it lived, and it grew. And I was like, oh my, this did better without me than it did with, like, how is that even possible? That's how bad of a, you know, horticulturist I am. But I enjoyed science, and I thought, man, maybe I could do this further in my life. So I figured, oh gosh, I'm going to apply to some graduate schools. I applied to some master's programs, and I ended up going to this school in North Carolina, in the mountains of North Carolina. It's in Boone. It's called Appalachian State, not Appalachian. If you say Appalachian, you're wrong. It's Appalachian. And this was our mascot. As a mountain guy who smokes a corncob pipe. Literally our mascot. It's like so stereotypical. But we were stereotypical. All we did was like drink moonshine and shoot guns and drink and shoot guns together, and rock climb, and do stuff that you do in the mountains. But it was a cool environment. It really put me in a position where I could spend time like actually doing science and learning about it and developing a passion for it. And I think that's the key, is like people view science as this like, thing you learn. And what I love about what Doug does here is that like science is a hands-on thing. Everybody's always asking me like, what book can I read to like learn about this stuff? Or like, what thing can I read? And I'm like, no, there's not a thing you can read. Like, you need to do it. You need to put your hands on this stuff and do it, right? That's how you learn. That's how you get that knowledge. And like some science concepts I could not understand, CRISPR including, until I just started working with it and saw the process and saw how it all worked. And then I was like, whoa, that's it. And I think that's a thing that people don't recognize. Everybody thinks science is like this intellectual thing that you just sit there and think and try to be smart. And that's what academia tells you, that like that's science, but it's really not. It's really not. It's doing stuff. So I did stuff and a lot of stuff, and I was like, oh, goddamn, all right, what should I do? Do I want to get a job or something like that? And I was like, I think I just want to, not work a job. So I applied to PhD programs, and I got accepted to the University of Chicago, where I went to graduate school. And it's an amazing university. I don't know if you all know much about the University of Chicago here. It's one of the top 10 universities in the world. But like most people outside the Midwest don't know much about it. I guess the microphone's cutting off the music and stuff, but we'll figure it out. Yeah, it was one of the like coolest moments of my life. I was just like, holy shit, this kid who failed high school and was arrested twice before he was 18 got accepted to this amazing university. I thought like, oh man, my life is like complete now. This is awesome. And I got there and I just dove right in and just passionate and working and doing research all the time. There, everybody worked on the weekends. You worked 7 days a week, 10 hours a day or more, and you just killed yourself for the science. That's what we told ourselves, like, yeah, we do it because we love science so much, but it really was the pressure. Everybody's putting pressure on you to perform, paying you like terrible, terrible wages, and, forcing you to perform. And I was, everybody tells you that science is like this meritocracy, right? You work in science, you publish papers, you work hard, and things will turn out well. And that's what I thought. And so I just, worked so hard, and this was my first paper, and I loved it. Like, the amino terminal helix modulates light-activated conformational changes in ASL2. Like, what does that even mean, It's so niche. It's just like, oh my gosh. But to me, I studied in graduate school protein thermodynamics, like how proteins move, and this was in relation to light. This protein absorbed a photon of blue light, ASLOVE2, and when it absorbed this photon of blue light, it moved. Like the protein actually moved from the energy that it got from absorbing this photon. And so what we did is we tried to use all these fancy techniques to see like how it moved, why it did a certain thing. So you could harness that movement to create little machines and stuff like that. At least that's what we told ourselves. But I worked so hard on this and so hard on my paper, and I created all these figures that I thought were so beautiful. I still, to this day, I'm just like, oh wow, you know, so pretty. And like, if you read this paper, like really read it, you can find out who I am as a person. It's just in there, right? The wording, the pictures, everything. But nobody else saw that. Like, we sent it off to a bunch of journals, and this commonly happens in science. It's not just me. But we sent it off to a bunch of journals and stuff, and it got rejected and rejected and rejected, and it was just like, what the fuck? Like, I thought if I just like work hard and do good work, that like things will work out and it'll be good. And it was ended up being published in this journal, the Journal of Molecular Biology, which is a great journal. You know, it's not a terrible journal at all. But nobody saw it the way I did it. Nobody saw the beauty in it, and I couldn't get that out of my head. Science wasn't like a passion for people. They didn't want to create beautiful things and see beautiful things, all these people that were around me. They wanted to publish papers. They wanted to get grants. They didn't want to do something that was just risky or crazy. And scientists, you know, academic scientists, they'll tell you, oh, you know, I'm trying to do risky and crazy things and stuff like that. But no, man, like, you know, we want dragons. We want invisibility cloaks, you know? That's what I'm waiting for. I'm not sitting here like, what's the light-activated conformational change of the amino terminal helix? I'll be the first person to tell you, you know, and everybody tries to comfort me when I tell them that, like, nobody is ever going to care about this research except for some niche, you know, people in the world. And everybody's like, oh no, maybe someday somebody will use it. And I'm like, no, it's not. It's okay, thank you. Nobody's gonna. But for me, I was just like, you know, what am I gonna do now? Academia is not a place for me. I'm trying to create these things I see in my head, these interesting, crazy things, and all we're doing is, you know, giving fruit flies cocaine and studying amino terminal helix. And so I decided to start just looking outside, and I started applying to government jobs and stuff like that. I wrote this crazy fellowship application for NASA, and it was like, yeah, you know, maybe we could genetically engineer some stuff and we could use it on Mars, and genetically engineer some more stuff and maybe use it on the space station or something. Like, it was really weird to write that and not like feel like you were being sarcastic or hyperbolic or something. You're just like, yeah, maybe we'll use this on the space station, you know. To me, it was pretty funny. Like, to write that seriously, I thought nobody would take me seriously, but they did, and they gave me this fellowship, and I worked down at Ames. I don't know if any of you have been down at Ames, down Mountain View, and I worked at the Astrobiology and Life Sciences Laboratory Centers for Nanotechnology and Mars Exploration. That was most of NASA, I'll be honest with you. They're just like, how can we make the biggest science possible and do the least amount of work possible? Let's make it sound really good, you know? Like, let's make our Twitter accounts have nice pictures, but the actual work we'll do will be really minimal. Got a good social media team, though, you know? Always posting those Mars rover pictures from last year. No, I mean, as much as it seems like I'm joking, I'm really not. Like, NASA was a crazy place where, like, nobody worked. Like, you'd come into work on any given day, and, like, I remember this one time, the fire alarm went off in our building, and, you know, the base, it's on a Air Force base and everything, so the base fire department came, and everybody came out. And there was literally, like, you know, this is a 4-story building and a basement, you know, probably housed, like, 40 labs or something, 30 labs, and get outside and there's like 6 of us. And the fire department's like, is this everybody? Seriously, firefighter says it's to us. And we're all looking at each other and we're like, this is the middle of a work week, you know, middle of the day. And we're all like, I think so, I don't know. You guys see anybody else? I don't know. I don't know what happened. And I'm not even, seriously, one time, one of my supervisors pulled me aside and was like, you know, you've been working too much in the lab, and if you work a lot in the lab, your chances of getting hurt are higher than if you just sit at your desk. So if you take like 1 or 2 days a week and just answer email and not work in the lab, that would be great. And I was like, what? I'm not even, this is just real conversation I had with somebody. And I was like, it doesn't make any sense. Like, what are you asking me to do? Not work? Like, I don't know. This is crazy. I got in a real big argument over that, which was like stupid of me. I should have just been like, oh, sure, you know, that's what I'll do, and then just go do whatever I want, because nobody's watching what I'm doing. But I just argued, and oh gosh, yeah. They were just like, oh man, this guy's trouble. So after I was there and I did a bunch of research working on, you know, engineering bacteria to break down plastics and break down waste and all this stuff. And I started using CRISPR and learning about CRISPR and all this stuff. And I remember learning about CRISPR and, you know, reading the papers. This was, oh gosh, when was this? 2015 or something like that. And there's no real good explanations online. There were all these acronyms. And it's typical, you know, scientist bullshit. And it's just like, the Cas9 protein binds the CRRNA and the tracer RNA, and then you have to find the PAM sequence, and it binds there near it, and the PAM sequence is sometimes NGG, but sometimes it's CCN. And I was just like, what is going on? Like, I don't even know, is that real words or something? And you tried to like look for the information in the papers and the supplemental material, but nobody would write about it. It was like they were purposely obfuscating this shit so other people couldn't, like, you know, catch on and like catch up to the research that they were doing. And I was like, holy cow, this is so complicated. And I started working with it, and I was like, God damn, this isn't that like, why doesn't somebody just explain it simply? It's not that complicated. I mean, it took me like a month, somebody with a PhD, like a month to figure this out, but like, it wasn't that complicated. And then I was just like, wow, science all across the board is pretty crazy, you know, like government, they don't want you to do work. In academia, they're just trying to obfuscate everything for everybody. It's a crazy world. And I remember coming into work one day, and this was like, I forget, it was maybe like a Wednesday, and I went around looking for people to see if anybody was around, just to bullshit with, you know, whatever. It's a real video. I don't know why I took it though. I was just kind of like fed up with everything, I guess. Yeah, you can see it's like 3 o'clock in the afternoon. That's the lab that Carl Sagan and one of his collaborators used to do some experiments and like messing around with creating like artificial atmospheres and testing the chemistry of all this stuff. And it was like, where is everybody? What's going on? Like, I don't know if I could do much more of this. That was kind of like, all right, I think I'm done. But what do I do next? Very dramatic, right? I probably did something really stupid. I don't even know why I did it, but I mean, what I learned in life is that usually if everybody's afraid of doing something, and I'm not talking about like, oh, jump out of a plane or something like that. I'm talking about like, if there is something that seems almost reasonable and nobody else is doing it, maybe because, you have to sacrifice a safe job or something like that. Usually it means that there's something there. Like, there's something valuable there because there's some force that's just like holding it back. And for me, that was science. It was like, I ask my friends even nowadays, I have so many friends who are like professors I went to graduate school with or met and working at NASA or all these things. A lot of friends who are still in academia. Most of them. And I ask them, I'm like, what are you doing? You have a PhD from a fancy university, and like, you're working a postdoc job making $50,000 a year in hopes that like science is gonna save you and save everything. Why? Why are you still doing this? Like, literally, you could be doing anything else. You could go work at a company for a few years, save up enough money to do whatever scientific research you want. And they're just like, it's like they're stuck in this system, and it's so hard to leave. It's so hard to leave because everybody's doing it. Everybody's doing the same science. Everybody's doing it the same way. But I figured, what if we do it a different way? What if people just like do science as a hobby? Do it in their home. Learn CRISPR in their home. That's what I thought. I'm like, I figured it out. Like, CRISPR isn't that complicated, and CRISPR could be done in people's homes or anywhere. Genetic engineering could be done anywhere. It's not that complicated. So I started making up kits and packing boxes and shipping out. First, we were in Burlingame, and I had this kitchen that was literally like 5 feet by 5 feet. And my partner who I was living with at the time, she was so pissed because it was just like a bunch of that I would just like pack up and send out to people. It was so fortunate, like the post office was literally like a block away. So every day I'd just pack up some stuff, walk down to the post office and send it out. And, we started selling a lot of stuff. And then we moved to Castro Valley. We upgraded to a garage in Castro Valley. Lived in the house, which was terrible. Don't live and work in the same place. Oh my gosh. That drove me to a lot of drinking. But, people were interested and people were buying this stuff and people were doing science in a different way. They were just like, what interests me? Not like what is going to sell or what is going to get me a publication, but what interests me? What do I think is cool or interesting and want to work on? And that's different for everybody, and it doesn't have to be something that's glorious and amazing. It could be the most pointless thing. In graduate school I worked on something pointless, but at the time that was beautiful to me. And there's nothing wrong with that, if that's what is beautiful to you. But, of course, when you start teaching people how to do genetic engineering in their homes, yeah, people freak out, right? They're like, what about eugenics? What about all this stuff? You're like, oh my God, people are just doing science experiments. Nobody's like engineering humans or embryos. It's just some science experiments in their home. People were freaking out. And it's, the way things are. People are afraid of this technology because there's so much you can do. It's so powerful, right? So we kept pushing forward. We started running classes, which you shouldn't take. You should take Doug's classes, We started a conference, which you should all come to. It used to be in Oakland at the Omni Commons, but this year it's actually going to be at the Golden Nugget Hotel Casino in Las Vegas. It's called BioHack Planet. A bunch of people who are doing science outside traditional environments, but also, researchers from universities. We've had plenty of researchers from universities come and give talks who are interested in people who are pushing the boundaries of this stuff. Because, You know, this is what makes science and CRISPR and genetic engineering so cool, is that people can do it. Because, like, literally when you think about the world, right? Like, these things are self-replicating matter. In genetic engineering, CRISPR, it's kind of like you're programming that self-replicating matter, right? So like trees, maybe you could imagine, you know, a crop that's as tall as a tree, right? This is matter that we can kind of engineer. Like, that's crazy when you think about it. The power that is, right? These little things, even animals, you know, they start out as these little embryos and they grow. Humans, we start out little and then we grow, right? Like, there's so much there. And that power, to be able to do that, to change those things, everybody should have access to it, I think. Even though it scares a lot of people, I think everybody should have access to it. Everybody should be able to genetically engineer. Everybody should be able to use CRISPR. And people agree, you know, lots of surveys that they've done have shown that people agree. People think that we should be using gene editing and genetic engineering to help treat disease. The majority of people, and that's just crazy, you know, that there's not more of this. And it's even more crazy is that, like, 39% of these people, this survey was done a few years ago, 39% of people are okay with, you know, somatic or like adult human enhancement, right? Like modifying your body to have enhanced characteristics. That sounds crazy. People are into that. People are okay with that, right? And it's, you know, kind of scary and kind of exciting. And I'm not the one who's going to be here and give you that Silicon Valley bullshit of like, oh, we got to go so fast and do all these things. Yeah, push through all the boundaries. But there are some huge problems that are going on. So there are gene therapies that you know, use genetic modification to help treat disease. The first one was called Glybera. It was approved in the EU in 2015, and they charged $1 million for it. Well, nobody could afford to pay for it, so eventually they took it off the market. This gene therapy is a functional cure for this disease, lipoprotein lipase deficiency. Sorry giving you all these hard words. But it's a functional cure for this disease. They took it off the market. And you know, people always have these conspiracy theories like, oh, I bet you there's like a cure for cancer that the government is hiding. There's literally a cure for this disease that nobody has access to. They just took it off the market because nobody could afford to pay. Imagine if the reason you were not getting treated for disease, you were going to die, was over $1 million. That's kind of f***ed up. And there's a treatment right now out called Zolgensma. It treats muscular dystrophy. And you have to give it to kids before they get around, I think it's 2 years old. Because usually the body starts to deteriorate rapidly. Usually die before you're 25. And not enough people can afford it, so they decide to hold a lottery. A lottery. Can you imagine that? If your child's life was going to be determined by what number you drew in a lottery, that's pretty f***ed up. The thing is that all this knowledge is available. You can go online and look at the patent and find the exact DNA sequence that they used to make Glybera. You can go and look at their, you know, regulation filings and find out exactly how they administered it, what dosages they used, what other drugs they give, what other things they monitored in the blood. You could give the exact same treatment and care for a cost that's in the tens of thousands of dollars. That's crazy. And this is why I think science, everybody should be learning about it. Everybody should be doing it. Because how are they going to stop people when they start pirating these drugs? Like, what are they gonna do? Oh, shit, you cured somebody? Well, we're gonna throw you in jail. Like, how long do you think that's gonna last? Right? Maybe the first few people they go after. And it's scary. It's scary for me, right? Because literally, I could do this tomorrow for somebody. Go online and order it. But it's against the law. I'll get tossed in jail. And I started working on stuff like this. I started working on gene therapies and how to work with them and understand them on a basic level. And the FDA put out a guideline. And they said, like, all right, self-administering gene therapies and all this stuff, we're going to strongly caution people against it. Journalists started writing about it. Biohackers disregard FDA warning. And Josiah Zayner. I don't know if it was the entire state of California. That's what Bruce Sterling said. Bruce. Ugh. And then I became a celebrity. They just wrote celebrity in there. I don't even know why. Like, what the hell? But yeah, like, I was under investigation for practicing medicine without a license from the Medical Board of California, and I'd never given anybody a drug, except that one time I tried to sell my friend weed in high school. But yeah, I never tried to sell anybody any drugs or give anybody any drugs or medical advice or treatment. So this was crazy. Like, people were coming after me for just giving people knowledge, telling them how to do genetic engineering, how to work with CRISPR and all this stuff. And then a senator down by LA made a law targeting my company. It doesn't make sense. The law is like, if you try to give people illegal gene therapies, you need to say not for human use on them. And you're like, what? Like, it's already illegal to give people gene therapies. Why it didn't make any sense. Literally named me and my company in like the f***ing bill. So I guess I got that going for me. And I was just like, oh my gosh, all I'm trying to do is teach people science. That's it. How come they're never coming after Doug? Like, he's got it easy. Maybe we look enough alike, we could be body double every once in a while. So I was like, what am I gonna do? And I was sitting down and I was watching the internet one day.
Back here live at the Waterfront Village with my friend, the zombie, Jonathan. You're looking good. Jonathan just got an awesome face paint job. What do you think?
I like turtles.
All right, you're a great zombie. And good times here at the Waterfront Village.
And I was like, damn, zombies are pretty cool, They just like turtles and stuff. And I'm like, maybe I could just turn everybody into zombies. And we'll all just like turtles. Nobody will harass me and all that stuff. And, I know all this CRISPR stuff. Maybe I could use CRISPR to do it. So, here's the part where you're going to have to maybe learn something. Oh, sorry. So, when I was learning about CRISPR, it was all these complicated things. But it basically came down to really two things. Two things make up CRISPR. It's this name that could mean a lot of things. And it's this enzyme called Cas9 and this piece of RNA called this guide RNA. And basically the way it works is this guide RNA binds to the Cas9 enzyme, and the RNA has matching base pairs, 20 usually, that's a good number to use to match stuff. And it finds the gene in the genome, and then the Cas9 enzyme cuts the DNA. That's it. It's that simple. Cas9, CRISPR, guide RNA, they don't do any genetic engineering. Contrary to popular belief, CRISPR doesn't do genetic engineering. All it does is cut DNA. And this has been something that we've known for a long time. So there was zinc finger nucleases before it. There was talons. Using things to cut DNA is the way, essentially, we genetically modify stuff. And why? Why does it work that way? It's because almost every cell in existence has a repair mechanism. When your DNA is cut or damaged, it wants to fix it. That's just the way it works. So like almost every cell has it. That's why CRISPR works in so many almost every organism CRISPR works in. Not every, but almost every. And it's because normal cellular repair processes, they are what cause the genetic modification. So what you do is you put CRISPR in a cell, and the Cas9 and the guide RNA come in the cell, and it cuts the DNA. Now there's two things that can happen when the DNA is cut. One is called non-homologous end joining, generally referred to as NHEJ. And basically what happens here is that when the DNA is repaired, sometimes it's faulty. So there's a mistake put in there. Now when there's a mistake put in there, usually the rest of the gene won't work correctly. So generally what you'll try to do is you'll try to cut in the beginning of a gene, right? And maybe some extra base pairs will get added, and that'll throw off the whole rest of the gene, and then that gene won't work anymore. Now this is good for, say, when you have a faulty gene or something like that you want to stop from working or knock out or remove. This is the main type of CRISPR that's used. So, contrary to popular opinion where you can like modify all these genes and insert all this crazy stuff using CRISPR, that's generally, it's a very low probability event. That's what's done through this other process called homology-directed repair. Now, homology-directed repair is the same thing. You get Cas9 in the gRNA and they cut the DNA, but instead of just letting it repair itself, what you do is you flood the cell with what they call a template. Now, a template is just, it has pieces that match the genome, and so the cell thinks this is a template. It's a copy of my genome. Hey, this looks like our genome. Let me use this as a template to repair myself. So it uses that as a template, and what you can do is you can like have little regions that are different. So you can say, take a gene that we want to insert in, and a region that matches the genome, cut the genome with CRISPR, and then it gets inserted. The problem is this is a really low probability event. So if you're trying to do it, say, in a human body, the ability to modify enough cells to be able to do this would be extremely hard. So it's already, hard to just cut the DNA. And you have to imagine, on top of cutting it, you have to have this template that's used to repair, right? And it's not always used. Sometimes it's just repaired normally. So generally, when people think about CRISPR and they imagine inserting all these genes into the human body and all this stuff, it's most likely not going to happen. It's very difficult to do on a large scale. So the majority of things that are done are, through this non-homologous end joining. But, we can just imagine, we're trying to create zombies. We can do whatever we want, right? So what I'm going to show you, it's really actually easy. You don't have to know much. And that's what's cool about science, is that other people have worked on this stuff a lot. So there are websites out there like Chop, and there's a bunch of other ones that you can literally just go to. You can type in a gene name, and it'll give you, the guide RNAs that will match that gene, right? You don't have to do any work. So, right, we know we need a guide RNA, we need a Cas9. Generally, your Cas9 enzyme is going to be the same DNA sequence all the time. You don't even need to worry about that, right? We got our guide RNA right here. So, if it was me, I would probably choose two genes. ApoE, which is associated with dementia and other stuff. So, maybe we could give people dementia. And then use this other gene, MAOA, which make them really aggressive. So the DNA sequence for a lot of this stuff is pretty standard. Everybody uses the same stuff. So you don't even need to worry about like all these things. What is all this stuff? I don't even know. CMV promoter, enhancer, NLS signals, what? All these things just tell the, how to express the Cas9 enzyme, get it into the nucleus, and how to express the guide RNA. So the next tough thing is, what virus do we use, So that's what's used for gene therapy, right? They actually use viruses just for standard gene therapy. Now, generally, the two main viruses that are used in gene therapy are the adeno-associated virus and the lentivirus. Now, some of these other ones are also used, but those are the two main ones. The two approved gene therapies in the U.S. both use what we call AAV, which is the adeno-associated virus. It's really not hazardous. It's a really great virus, doesn't really cause much of an immune response. But the problem is its genome size is really small. So it's got about, 5 kilobase, that's KB, so 5,000 base pairs of DNA it can hold. And the smallest Cas9 size we have is about 3.2 KB, right? So you're talking there's not much room for a lot of other stuff. So it's difficult. You can fit the Cas9 to guide RNA, but say we want to throw in a template, we probably want to use like a lentivirus or something like that. The problem with lentivirus is it incorporates into the genome a lot more often. So people are kind of afraid to use it as gene therapies, but we don't care in this case, right? We want it to get incorporated in people's genomes because they're just going to be zombies anyway. They don't care. So I'm thinking we go with the lentivirus. That's probably going to be pretty good. So what you can do actually is not that crazy. So there are companies, I'm not going to say this company's name even though it's in the URL. Companies hate me. I actually had a company, what was it? Was it ADAM, which used to be DNA 2.0? We had a guide on our website of how to create CRISPR for human genetic modification. Just a guide written up. And they emailed us, their legal team emailed us and said, can you please take that down because we don't want our name associated with you. And I was like, I don't know. Like, what are you going to do? It's not illegal for me to have a link to your website on my website. They didn't like we were promoting them as a way to make your own CRISPR. But there are companies like this which you can literally order your own AAV, adenovirus, lentivirus, or plasmid with Cas9 and gRNA together, right? They'll do all the work for you. All you have to do is literally give them the gRNA sequences from ChopChop, right? So just go to ChopChop and be like, all right, I'll use this target sequence, and I'll use, this target sequence. Can you make that for me? And it'll probably be a few $1,000s, maybe like $10,000 for those two viruses, But then we're ready to go. So, I was so tired of the world harassing me. That's what I decided to do. I just decided to make those viruses, and that was it. The zombie apocalypse happened, or maybe it didn't. We're all here still, right? But this stuff is so easy, right? And it's just like this lack of knowledge. It's this obfuscation of knowledge that happens that makes this stuff not accessible. But literally, in the past 10 or 15 minutes, you just learned how to do CRISPR, right? And it's not that much more complicated if you already know how to use a pipette and all this other stuff. Like, you could learn to do CRISPR with Doug. You could learn to do CRISPR anywhere, right? I think this is a zebrafish embryo, maybe. So, in November 2018, I don't know if many of you heard of this, but there was a scientist who genetically modified some human embryos with CRISPR, like for real, and implanted them in a human being, and they came to term. For all of human history up till that point, every single human being, their genetics were determined by stochasticity, pseudo-randomness, who their parents were. but nobody has been able to choose what genes and genetics they have, or choose the genes and genetics really that anybody else has. But right now in human history, right, we can actually modify human genetics, we can modify plant, animal genetics, all the genetics you can imagine. But what we're limited by is we're limited by the number of scientists and researchers. We're limited by the number of people who have knowledge and skills to be able to do this, right? And I'm not saying like, oh, don't go get a job, because like we all need f***ing jobs, you know? Like I work a job. I don't just sit at home and tinker with DNA stuff. What I'm saying is like, if you learn this stuff from Doug or whoever, like use it in a way that is beyond what people normally use it for. Express yourself. Create something cool. Create something beautiful, right? Because we are at a crazy moment right now in history when it comes to genetics and genetic engineering. And you can be part of that, right? You can learn to do CRISPR hands-on. I mean, basically just learn to do it, right? Right there. You can order it. You can be part of this whole thing. And that's what I wanted to tell you today, is that like, not just CRISPR, it's not just genetic engineering, it's all of science is becoming accessible to you. And amazing people like Doug are making it super accessible for you. You have to take advantage of stuff like that. You'll never have a better chance in your life, right? It doesn't matter what's going on, how old or young you are, right? You can contribute, you can do cool stuff. But that's it. Thank you all for coming.
Great. Thank you. Yeah, there's some time for questions. I'm sure there's lots of them. So I just wanted to just say everybody speak up, and if you don't mind repeating the question too. So
I'll do my best. Don't ask me if it will cause a zombie apocalypse. It's a good question. So he asked, what if somebody does something wrong with this stuff? You know, it's totally possible. Like with computers, he was saying that like people created computer viruses and they were bad. What I would say is that genetic engineering doesn't have intent, right? It's really hard, almost impossible to do something wrong on accident, right? Just like computers. You're not going to like write a virus on accident or anything like that. It requires intent to do it. Now, everything when looked at in that way makes you think like, well, what about a pressure cooker? Do we not give people pressure cookers because somebody could turn into a bomb? Or like automobiles. Automobile can be used as a weapon. In fact, over a million people a year die in automobile accidents. We sacrifice them to the gods of automobiles. And if you ask somebody, should we get rid of cars? Nobody will tell you yes to save lives, right? So I think it's this risk-reward thing. Is something bad going to happen? Of course, like people are going to use all technologies for bad things, I think. I think the question more becomes like, does the good outweigh the bad? And I think the answer is undoubtedly yes. There's so much good that can come from people having access to this technology. I think the majority of it will be good, just like computers, right? If you were to say, should we not give access to computers and programming to people? I mean, Linux, right? The operating system that runs the world and runs our Android phones, everything, was written by Linus Torvalds in his apartment in Finland, right? Because he had access to the technology. And if we wouldn't have given him access to it, where would we be today? I don't know. But that, you know, you can have a different opinion. I'm not saying my opinion is correct, but that's just the way I feel about it. What would I engineer about myself? You know, I think wings would be pretty cool. That would be a big one, you know. I think that would be really cool to have humans with wings flying around and stuff. I don't know, you know. I think this is going to be an interesting question for the future, especially because people are going to get to make weird choices about themselves, you know. Like, choose their skin color. And I'm not saying that in a bad way, but like, what if you could be like, you know, blue or green or something like some people want to be. That's going to be weird, but hey, you know, whatever you want in eye and hair color and other things, like, it's going to be a weird place. But I don't think it's going to, like, push itself to homogeny. A lot of people think, like, genetically engineering humans is going to cause eugenics, and, like, I think when the government controls things and forces you to do stuff, that's eugenics. But when you start to be able to express yourself, like, there's going to be no homogeny. It would be the furthest thing from homogeny we can imagine. And the other thing is, I definitely would give myself a gene therapy so I didn't have to go to the gym. Just be able to eat whatever I wanted. Like, damn, I'm getting older, you know? It's getting hard. Yeah, so question was asked was like, an embryo or a person that's not yet born, they can't consent to changes that you might make to them genetically. Like, as an adult, you can consent to changes you want to make to your body, and that's cool, but like an embryo can't. It's a tough question, and I don't think there's a correct answer for everybody or every country or every society. But what I think is that, like, children never have consent, you know? Like, I have a daughter who's 2 years old, and she got vaccinated, and she had no say in that. You know, like, she gets to eat what I feed her. She gets everything about her life and her health is determined by me, and it will be probably up until she becomes, like, you know, a teenager, when she could start making rational decisions for herself. And so for me, it's like, you know, and even before that, you're talking about, we already select for genetics in embryos, right? It's not genetic engineering per se, but we screen for different things in embryos, right? Like, you do IVF, and they give you a list of, here's your eggs, and 3 of them had bad genetics, so we won't use those, right? 3 of them had negative mutations. Now, if you couldn't tell the difference between a negative mutation and a CRISPR genetic modification, which is totally possible, you wouldn't be able to tell the difference after the fact. Is there any difference? That's a tough question, right? Selecting for a gene versus modifying the gene. Yeah, I don't know. And, like, I don't think there's a correct answer to that or any of this stuff. It's just purely my opinion, and I think there's so much more that goes into play sociologically, religiously, and everything. And it's complicated.